Hello, my dear student. Welcome to UJPharmaAcademy. Please like the channel, like the video, and subscribe this channel. Uh, Today, I am going to start research and methodology of the CM pharmacy. So now we have to learn and this channel. So now we start the unit number 2, biostatistics. In that biostatistics, in first one, अगर एज पर द पीसीआई आप अगर सिलेबस देखेंगे इन बायोस्टैटिस्टिक्स फर्स्ट उसका डेफिनेशन है एप्लीकेशन सैंपल साइज एंड इम्पोर्टेंस ऑफ सैंपल साइज factors influencing the sample size, dropouts, statistical test of this significance. There are in two types of this statistical test of this significance. One is the parametric test and another is the non-parametric test. In first one is the parametric test is the student t test, ANOVA test. In ANOVA it contains two types, ANOVA 1, one way ANOVA and two way ANOVA. So next third one is the correction, correlation coefficient and regression. And non-parametric test is Wilcoxon rank test and analysis of this variance, correlations, Chi-square test. So comma then null hypothesis and p value, degree of freedoms and interpretation of this p value. So अभी चलते हैं कि वन बाय वन हर एक चीज को पढ़ते हैं. सभी को मेरा नमस्कार और वेलकम. सभी को आप अच्छे से इस यूट्यूब चैनल से आप पढ़ सकते हैं रिसर्च मेथोडोलॉजी यूनिट नंबर 2. So first of all we have to know that the in basic of the biostatistics is the definition is the biostatistic is the branch of the statistics that deals with the study of the application of the statistical method to problems in biology, medicines, pharmacy and public health. So what is the preventive? In this pharmacy and medicines it helps in drug discovery, clinical trial, epidemiology and equivalence studies, bioequivalence studies and evidence based decision making study. Then is the you have to provide the tools for the collect the information, organize it properly, then analyze the data and interpret the data, and presentation is ongoing. So and this applications of this biostatistics, what kind of these applications used in this biostatistics in different field. So first one is the clinical research. In clinical research is the designing of this clinical trial, sample size determination and analyze the efficacy and safety of this of drug molecules. The second one is the epidemiology, studying incidence and prevalence and spread of the disease. And in pharmaceutical science is most important aspects regarding to the some kind of this pharmaceutical students in the formulation development, stability studies and quality control. And the fourth one is the public health, evaluating the vaccination program, assessing healthcare policies. And fifth one is the genetics and molecular biology, is the genome studies and bioinformatics. Then most important aspects to the sixth one, regulatory submission. In that regulatory submission some of the students are coming from this in the CM pharma in regulatory affair. That time the regulatory submission is the very most important, is the statistical evidence required for the in this food and drug administration as well as EMA and CDSCO approval. CDSCO is the national regulatory agency of this India. CDSCO is the Central Drug Control Standard Organization. In that move towards the first one is the sample size. Definition: the number of participants, comma, patients or the experimental units is the select from this the study. And what is the purpose of the sample size? To ensure the results are valid, precise and generalizable. And this formula of the sample size is simplified from this proportion of formula for the comparing the proportion in this clinical trials. If the formula is n is equal to z square into p into 1 minus p divided by d square, where is the z is the standard normal deviate, P is the expected proportions, and D is the allowable errors. Next one is the formula for the comparing of these two means. Okay, n is equal to 2 into z alpha by 2 plus z beta is whole square into the gamma and d square. And next move towards the importance of sample size. In importance of the sample size, first one is the ensure the adequate statistical power to detect the true differences and reduce the chances of type 1 error and type 2 error. Third one is the prevent the overestimation or underestimation of this treatment effect. Fourth one is the balances the accuracy with the resources like time, cost, and manpower. The what kind of these factors are influencing the sample size? In first one is the study objective. In that, the exploratory studies may use the smaller samples, and confirmatory trials need large samples. And second one is the types of the study design. Okay, in that the parallel study is there, crosswire study is there, and the case control and cohorts. The effect of this effect size. In the large effect requires the smaller sample, and small effects require the larger sample. In that variability in population, if the higher variability, you have to get the large sample. Sorry. Fifth one, significance level is alpha is commonly 0.055% risk of this fails or positive. And sixth one is the power to predict the to predict the detecting true effects. Fourth one is the seventh one is the dropouts, and is the also known as the non-compliance rate. Is the researcher add the 10 to 20% more participants to compensate. In that most we have to go through the what is the some of these examples to survey in this pharmacy practice. Example 1, suppose a pharmacist want to the study the proportion of this patient who adhere to their antihypertensive medications. In that farm, the given data from the past studies is adherent rate P is equal to 0.7 as well as the 70% confidence level. 95% Z is equal to 1.196, and available error D is equal to 0.05 as well as 5%. You have to calculate there. In that, in the n is equal to, you have to put the values on this for the given formula. Then you have to calculate the how many patients are required. These are the samples of this sample size analysis of this supposed to examples. Example 2, a researcher want to compare a new anti-diabetic drugs with a standard drugs. So if the given data is there and expected differences in this HbA1c reductions, and between two groups is 1% standard deviate, significance level, and power. So these are the formula for the comparing two means, and we have to put the value of this particular formula and calculate the sample size. Next, move towards the most important points to the dropouts in clinical and pharmaceutical research. The definition: a dropout is a study and participant who fails to complete the study as per the given protocol. And this means the subject either the withdraw voluntary, is the lost follow up, or is excluded for this medical and ethical reason before the study ends. Then this is the dropouts statistical, and what kind of this causes of these dropouts. Just we have to see here the causes of these dropouts. First one is addresses the effect of treatment. Is the patient discontinue due to the side effects, toxicity or intolerance. Second one is the lack of efficacy. If the patient feels no benefit, they may stop participating. And third one is the personal reason, the lack of interest and migration, family commitment, and job relocation or time burden. This is the personal reason. And the fourth one is the medical reason, the hospitalization of this morbidities or the death. So Fifth one is the non-compliance, if the patients not follow the prescribed treatment schedule, and sixth one is the is totally depends upon the study design issues. And two, many visits of this invasive and procedures or the long study duration. These are the uh, causes of dropouts. नेक्स्ट अगले वीडियो में मिलते हैं दोस्तों, अगले वीडियो में मिलते हैं। बस आपको लगे रहना है और आपको पढ़ाई करनी है। थैंक यू।